Okay, our last section deals with enzymes in medical applications and disease. Certain enzymes are normally found almost exclusively inside tissue cells and are released into the blood and other biological fluids only when these cells are damaged or destroyed. And because of the normal breakdown and replacement of tissue cells that go on constantly, the blood serum contains these enzymes, but at very low concentrations. However, blood serum levels of cellular enzymes increase significantly when excessive cell injury or destruction occurs, or when cell growth occurs rapidly as the result of something like a cancer. So, changes in blood serum concentrations of specific enzymes can be used clinically to detect cell damage or uncontrolled growth, like in something like a cancer, and even to suggest the site of the damage or a cancer. Also, the extent of the cell damage can often be estimated by the magnitude of the serum concentration increase above normal levels. And for these reasons, the measurement of enzyme concentrations in blood serum and other biological fluids has become a major diagnostic tool, particularly in diseases, diagnosing diseases of the heart, liver, pancreas, prostate, and bones. In fact, certain enzyme determinations are performed so often that they have become routine procedures in the clinical chemistry laboratory. And the table that you see here lists some enzymes used in medical diagnosis. And so now that we've finished the chapter, you should be able to identify alpha amino acids. You should be able to draw structural formulas that represent various ionic forms of amino acids, whether they're in strongly acidic solution or strongly basic solution. Right reactions to represent the formation of a peptide, so the formation of an amide bond between amino acids, and the oxidation of cysteine. That's where we take two thiols and they come together to form a disulfide, right? It's oxidation because you're losing hydrogens. Explain what's meant by primary structure. So remember, primary structure was just the amide linkages between amino acids, peptide bonds. You should be able to explain a little bit about secondary structure and hydrogen bonding that takes place in secondary structure. So alpha helix, beta pleated sheet, that kind of thing. Next, describe the role of side chain interactions in tertiary structure. So hydrophobic interactions, hydrogen bonding, salt bridges. Then you should be able to explain what is meant by quaternary structure. So when we have two or more subunits that come together to make a functional protein, describe four biological functions of proteins, three reactions that enzymes catalyze, describe enzyme activity, the terms of efficacy and mechanism of action, identify four factors that affect enzyme activity like pH, temperature, etc. Compare the mechanisms of competitive and non-competitive enzyme inhibition and allosteric regulation, and name three enzymes that could be used in diagnostic testing to identify the presence of a disease.